everything about the IND-2001d circulation.
Excellent Polish, Mr. Van Manen. So my talk today relates to full genome study on evolution of the India 2001 lineage of foot-and-mouth disease virus, and I'm going to show evidence of recombination within the lineage as well. So there are two approaches to sequencing of foot-and-mouth disease virus. One is looking at a partial sequence, sequence encoding the outer capsid protein VP1, and this is only about 8% of the full genome of foot-and-mouth disease virus, but it is still a very, very powerful tool for monitoring outbreaks, tracing transboundary events, as well as categorizing field strains. Yet, there is also the depth of the data that had been collected over decades and which is available either publicly or within different repositories. The other approach is looking at the full genome, full length of the genome. And obviously, the more sequence data there is available, the better the resolution of the results. And within the WRL at Pirbright, we have got experience of working with full genome sequence data. We have published papers on reconstructions of foot-and-mouth disease movement at different scales, reconstruction of farm-to-farm transmission events during the 2001 and 2007 outbreaks. We have also looked at reconstruction events, transmission events in Bulgaria in 2011. But the virus today we're concerned about is a serotype O virus, and within this phylogenetic tree shows the diversity within, this is not what I wanted to show, within serotype O with 11 topotypes. India 2001 lineage is within the Middle East, South Asia topotype, and this topotype had been described to circulate within the Indian subcontinent. It was first described in 2001, and it was shown that the lineage divides into 4 different sublineages. Perhaps the names of them not very inventive. They are sublineage A, B, C, and D. And indeed, the D one is the one that we're concerned about at the moment. So the area of circulation of the virus is within the Indian subcontinent, but there is perhaps difficulty to access the sequence data or indeed the viruses from this geographical region. But what we can do is to somehow zoom out and look at the circulation and evolution of the virus from outside of India perspective. And indeed, there were outbreaks of all the different sublineages outside of the Indian subcontinent over the years. And the first outbreak of India 2001 lineage happened in Iran in 2001. I have plotted almost 350 available sequences that were either publicly available or sequences generated at the Pirbright Institute and into a phylogenetic tree. And I have also listed countries that have been affected by the virus over almost a decade. And when you plot or project outbreak events into the topology of the tree, into the scaffolding of the tree, one can see that some of the events actually cluster together. Some of the outbreak events cluster together. This perhaps indicates that there is a lack of data, intermediate data, that could be available to us to somehow show the closer link and the recent links with the outbreak events. I have selected 56 viruses for full genome studies. And it won't be a surprise to you that perhaps a majority of these sequences were indeed from the outbreaks viruses that had been submitted to the Pirbright Institute and therefore available. I've used the Illumina MySec technology to look at the data and used a published protocol. So when Bayesian evolutionary analysis are performed on the data, there is perhaps a lot of things that we could conclude from the data, but what becomes very apparent is that majority of the samples that we can analyze are actually from 2013 onwards. So not only we've got gaps in knowledge in terms of geographical location of the samples, but we also have got gaps in terms of time scale. However, still, looking at the analysis which trace their most close related common ancestor on the time scale, the analysis indicate the origin of the outbreaks within Indian subcontinent. There are two major groupings within the tree, and so two major clusters, and viruses that are represented in both of the clusters were collected from outbreak events. So here we can see outbreaks in UAE, Bahrain, Laos, number of outbreaks to Sri Lanka, then further on outbreaks to Saudi Arabia, and indeed the outbreaks in North Africa, which Julia is going to talk about in detail later on, but we can see evidence of the introduction of the virus in 2013, and then the movement of the virus to Tunisia, Algeria, and finally to Morocco in 2015. The second cluster relates to perhaps newer events. And the outbreaks in 2016 in UAE, Saudi Arabia, two outbreaks, perhaps two introductions in 2016 and 2015, and perhaps of the press sequence data from Mauritius and a small island of Rodriguez, a small separate sort of clustering in this cluster. So we can see that really only recently this small cluster has arisen and starts to populate with phylogenetic events of evolutionary events. So if we compare the VP1 tree with the full genome sequence data, we could also see, perhaps not that clearly, but there are also two major clusters of viruses within the VP1 sequence data, similar to here, but also the smaller clusters, the subclusters, are also reflected on the VP1 tree. This perhaps indicates the power of the short fragment of the genome and the usefulness for tracing of these events. There is, however, one virus that doesn't seem to follow the pattern, the general pattern, and this is the Bahrain virus 15, some virus from Bahrain from 2015. And this virus not only doesn't fall into any of the clusters, doesn't group together with any of the other viruses, it doesn't also group with the other virus from Bahrain, which was isolated at more or less the same time. This is indicative of recombination events, and indeed we have performed some data using simplot analysis, which this data, we used 3 different viruses, 2 Bahrains, and 1 virus from UAE. When these compared, it shows that the capsid coding region is highly conserved between the 3 viruses. However, in the structural coding region, the Bahrain virus varies from the other from the other two, indicating recombination. We do not have the sequence data that we could identify this sequence, but perhaps this has originated from the Indian subcontinent. We have plotted, again, a Bayesian tree looking at, based on VP1 sequence, and indeed, as one would expect, the two Bahrain samples, two Bahrain sequences appear to be very closely related. And the overall topology of the tree also remains the full genome sequence data very closely. This is another type of analysis that we have confirmed called a TCS analysis based on the full genome sequence. And this type of analysis is very useful for tracing outbreaks and is based on the nucleotide differences and the quality or character of these differences between samples. In this projection of the tree, the length of branches is not proportional to number of nucleotide changes within the sequence. However, the major groupings as compared to the whole genome sequence tree remain the same. So viruses group together, sequences group together similarly to the full genome sequence data. And indeed, we can see the Mauritius, the events in Mauritius grouped together with the recent events in Saudi Arabia, UAE, but are also linked to viruses in Nepal and Bangladesh. I think this is right, Bangladesh. So really, this is my last slide, and this one you have seen before during the talk, and this picture represents the Indian subcontinent, and it shows that really this is the main area of circulation, and this picture really asks a number of questions. Why is this virus so dominant within India, and how does it happen that it escapes on so many occasions, and these events of escape do not seem to be linked together? So, leaving you with these questions, I would like to acknowledge our collaborators for providing samples and sequence data, in France, ANSYS, India, Italy, Thailand, as well. Nick Lyons has provided very useful connection with Saudi Arabia. I would also like to acknowledge all local and reference laboratories who have contributed to the work. Thank you for your attention.
Thank you very much, Kasia. Beautiful pictures and graphics. Questions? I have one rather practical question. Why did you take 56 viruses for the whole genome sequencing? Was that a pragmatical approach, funding available, or what?
Well, this is an ongoing study, so obviously the more samples that are available to us, the more sequence data we can produce, and obviously we encourage people to share the samples and share the sequences, so therefore we could extend the number of sequences that we have analyzed and perhaps provide a better data.
Okay, thank you. Any other questions? No? Thank you very much again.
Thank you.
So now we will travel to Pakistan.